So it's a pleasure to be here. I'll be talking to you about some very applied work. My work is very applied, and it deals with analysis of large data that is biological data. And this is joint work with Gunnar Carlson, with a lot of people actually, but the primary collaborators are Gunnar Carlson and Sylvia Plevritis. Everyone knows who Gunnar is, I'm sure here. But Sylvia is an engineer and a computational biologist, and I'm a mathematician and computational biologist. So let me start with some ancient history. So in this world, 20 years ago is almost bordering on prehistory. So things move very, very quickly in this field. So the Human Genome Project was started, and the idea was to find all the genes in the human genome. And the number of genes changed between 20,000 and 40,000 and oscillated back and forth. Now we know that they're about in the low 20s, low 20,000s, that is to say, of genes. So this is very, very large data. And large data doesn't mean there's a lot of computations that you have to do. It actually means a lot more than that. The nature of the data is very, very different. And that makes the field very beautiful for computational people, because we have to sort of rethink a lot of things. So first of all, it's a very new thing. Not in this field. 20 years is a long time, but in terms of the way we think, what we know how to do, it's a new thing. We couldn't generate this kind of data without these guys, yeah? And you couldn't do anything. You couldn't store the data. You couldn't look at the data without these guys, right? So it's a new thing. And so we have these problems that were sort of around before, but they meant very different things before. So what does it mean to look at data? Oops, too many buttons here, but I'll get used to it. There we go. What does it mean to look at data when your data is in 3 dimensions or 5 dimensions? You know, maybe you could do that. And what does it mean to look at data when it's in 20,000 dimensions? What does it mean to find patterns in your data when the data points are so far apart because the dimension is so high that, you know, you're bordering on, you know, connecting dots between stars to see that there's Orion over there? It probably doesn't have very much meaning that there's Orion. So you want to find patterns that actually have some meaning. Even something like computing something. How sure are you that what you're computing is actually correct? If you do it on a piece of paper, several other people can look at what you did. They can recompute, and hopefully, you know, you can be pretty sure that the computation is correct after a while. If you do it on a computer, you're relying on all sorts of algorithms. And there are people that will tell you that essentially there's no such thing as code that has more than 3 or 4 lines that doesn't have a little bit of bugs in there. Okay? So there are a lot of, I'm not going to give you a talk on all these things. I just want to say that all these things, all these concepts that were around before are very different now in this context, in the context of large data. The 2 things that I think about the most are these 2. The concept of relevance is very interesting, very difficult now, so therefore very interesting, and the concept of the shape of the data. So let me start with this a little bit. The shape of the data is topological data analysis, which is something that you've heard probably various people talk about, especially Gunnar Carlson. And shape of the data is basically intrinsic to any kind of data analysis. If you talk about outliers, you're talking about things that are far away from the bulk of the data. If you talk about the mean of the data, the center of mass of the data, you're talking about the stuff that's sort of most prevalent in the data. Yeah. So those concepts are concepts that were around all the time, and they're concepts about the shape of the data. But what does it mean to talk about shape of data when you have data that's sitting in 20,000 dimensions and you have just tiny little points? It becomes much more complicated. The issue of relevance, and I specifically use the word relevance, which is not a precise term, rather than the word statistical significance. It's also a very, very difficult concept now. Why? Because this data, and you'll see in a minute, this data really contains tremendous amount of information, most of which is irrelevant to the specific question you're asking. And what you have to do to the data to extract the part that's relevant becomes very complicated, because you have to squish the data around, do geometric transformations on the data to highlight things that are relevant to a specific question when you have a data set that can answer many, many different, very, very distinct questions. The same data set contains information about many different questions. So how do you extract the part of the data that's relevant to a question and still take advantage of the fact that you have very large data? So you'll see in a minute what I mean by that. I'll keep coming back to this a little bit. So the Human Genome Project was started, so people were very excited that they were going to find all the genes. And they very quickly pointed out that simply finding the genes doesn't really solve the problem at all, because we have to know what these genes do. I mean, otherwise we just say George, Fred, and Mary are the genes, but we still don't know what they're doing. So understanding what the genes do is a very difficult problem. And very soon people came up with all sorts of technologies to get information about these genes. And I'll just be talking about one of them, and I won't really go into the actual technology very deeply because it's not really relevant over here. Gene expression microarray. So the idea is to measure for a tissue sample, for example, tumor, the level of activity of all the 20,000 genes at the same time. So in other words, you get the data vector for this particular tissue sample is a vector in art width, not exactly 20,000, but something like that. The coordinates are the genes, and each entry here is an indicator of just how active that gene is. So in other words, some genes are sitting quietly and they're not being copied. The proteins that correspond to those genes are not being made, and some genes are being copied a lot, and maybe the proteins that correspond to those genes are then being made. Okay? Not necessarily, but that's the idea. So that's what I mean by a gene being very active or very inactive. People started looking at tumors very much. People always looked at tumors because you want to study disease. But in this particular case, it's very nice to look at tumors because because you can't do experiments on people. So we want to find out what's the function of this particular gene. You can't actually go up to someone and just shut down the gene because they're probably going to die, or maybe nothing's going to happen actually. But yeah, obviously you can't do that. But cancer sort of provides those experiments, right? So cancer messes around with the genes and changes their level of activity. So if you look at the tumors, you actually get an indication. So it's not just because you want to help people with cancer. It's also because you actually get to learn a lot about what these genes are doing by looking at tumors. So, so the data matrix, so the data is put together into a matrix, and data matrices are not matrices in terms of, as far as linear algebra are concerned. They're just sort of storage places for the data. I mean, you don't multiply data matrices together. You don't think of them as transformations on Euclidean space or anything like that. So, so the data matrix looks something like this, where there are about 20,000 rows, or more actually, and a few hundred columns. The columns correspond to the tissue samples, the individual tissue samples, like tumor 1, tumor 2, tumor 3, normal tissue 1, normal tissue 2, so on and so forth. And the genes correspond to, the rows correspond to genes. And this is an example of high throughput data. Okay, so high throughput data is data where you essentially query each object, each tissue sample, for example, along a ton of pieces of information, most of which is completely irrelevant to what you're going to study. And why is it good to, why would you want to do this? Because people knew, so let's say people knew that there were these handful of genes that are involved in this particular pathway, let's say. Biologists knew this. But what if there are some other genes that are also involved in this, maybe in an indirect way, or maybe they just weren't noticed? So the treasure in this kind of data is that it gives you information about everybody, so that you might be able to find things that you never knew to look for before. On the other hand, there's a lot of information here, and most of which is irrelevant to what you're trying to study. If you're trying to study this particular pathway, for example, right? So there's a lot of other stuff going on. So let me give you an example. If I want to find out structure in the people in this room, I want to give you all a form, and you're going to answer lots and lots and lots of questions about yourself. Things like, what do you do when you're reading stuff for work? What's your favorite color? Where did you go? Where did you spend your vacation 5 years ago? Where were you born? What do you eat for breakfast? Random information like this. Lots and lots of things. Because some of it might have something to do with what I'm interested in, which might be something about your activities when you're relaxing after work, for example. Okay? But there's a lot of other information there that has nothing to do with what you're doing when you're relaxing after work. So I have to somehow find a way to extract what is relevant out of this sea of information, most of which is irrelevant, without deciding a priori that this is relevant and this is not relevant. So that's a difficult problem, and I want to approach it sort of like a Martian. I don't want to assume ahead of time that what you do in your spare time has nothing to do with the kind of food you eat for breakfast. Because maybe it does. Probably not, but you never know what happened. Okay. So the main steps of the work I'll be showing you are something like this. First, you start off with data, and you do some kind of geometric transformation on data. So these are points in R20,000 or Rn in general, right? And I want to squish them around so that I highlight somehow information that is relevant to a specific question that I'm asking. So that's the geometric transformation of the data. And then I want to look at the shape of this data set after it's been squished around to see characteristics that perhaps have something to, shape characteristics that have something to do with the question I'm asking. So for example, if I see something that looks vaguely circular, maybe I'm looking at some kind of recurring pattern, recurring behavior. But maybe it could mean something else. It could be that a circular pattern might have something to do with the fact that there are two ways of getting from A to B, right? This way and that way. Or it might have nothing to do with anything, even though that showed up. So I'll give you an example. So the step 3 is to actually, and I won't really talk too much about that, is to interpret what you have. And then very, very importantly, and something that mathematicians always need to remind themselves, even when everything makes beautiful sense, you still have to go to the biologist or the scientist and ask them, is this correct? And very often, it's completely wrong, because it happens that nature doesn't care about theories, doesn't care about any of these things, and it does what it wants to do. So, okay. So I'm going to show you some pictures. I don't know how well, hopefully you can see them. They're just sort of mathematical teasers. I mean, so this is sort of made up pictures to kind of highlight issues that you want to look at. So let's suppose I have data points like this. I want to notice that this is a circle, vaguely. Well, if I have points like this, I have these outliers, so maybe I want to dampen them a little bit. It might be that actually the outliers are the interesting ones. But let's say that they're not the interesting ones. They're just sort of noise or not relevant to the main thrust of the data. I might have more outliers. You notice the point here, I hope. More outliers. It gets harder and harder to figure out what to dampen. The dampening is sort of a geometric transformation on the data. I want to push those guys down. What if I have this? So this is important. Okay, so what if I have this? I want to see this. Yeah? But it could happen that this part, the green part, is the only part that is related to, that's associated with the biology I'm trying to study. This other part is also a pattern, very clear pattern. These are not outliers, but perhaps they have nothing to do with the question I'm asking. So the pattern is in the data, and if I ask a different question of the data, this might be very relevant to that question, but not relevant to the first question, the one I'm studying. So if that's the case, I want to dampen these guys, and I want to see this pattern. But maybe the opposite happens when I ask a different question of the same data set. I want to dampen the guys in the middle and see the circle. And now, finally, look at this situation. That's a cylinder. I mean, this is the best I can do as far as artistic prowess is concerned. Okay, so this is I take the cylinder above these points that lie kind of in a circle, and the data sits along the walls of the cylinder and at various different heights. Okay, so the data points are these pink guys. So imagine that I'm looking at particles moving around in 3 space, okay, and their movement is influenced by all sorts of different forces. But I'm not interested in all the different forces. I'm only interested in one force, or a collection of forces, that cause this circular pattern, okay? So in essence, I want to push the points down so that I ignore this vertical component of the data so that I see the circle, okay? And now you might say, well, why do I have to push it down? Why don't I just notice that this is a cylinder and just ignore the height? Well, first of all, ignoring the height is exactly what I'm talking about. But the point is there could be all sorts of very disruptive, confusing patterns going on up the walls of the cylinder, right? So there might be all sorts of other circles along the walls that come from all sorts of other issues that are happening, which I can't control, I can't quiet down. So I want to quiet them down mathematically. So that's sort of the idea of the geometric transformation. Clearly, it looks like the more stuff I add to this, the harder the problem is going to be. So I might be introducing all sorts of problems, but they're not really doable. So let me show you a real live data set, not biological. And I'm going to show you that it's much, much more complicated than this. And your brain can deal with it. So if your brain can deal with it, then it's our job to figure out, and very often it's our job, I think, to mimic what the brain is doing so that we come up with a method to deal with this. So look at this picture. The older people in the room might recognize this. But, okay, so I don't know if it's dark enough for people to actually see the details of this picture. Anyway, do you see the lion's head? This is a lion roaring, right? Okay. So those are the fangs and the nose and the eyes, right? And the whiskers. Yeah, much better. Okay? So that's a pattern. And your eyes can distinguish between the lion's head, the mane, right? There are, I don't know what, millions of points in here. Lots and lots and lots of points, right? You can find that pattern. Now, look away from this for a minute. Unless you know this picture, you don't have to look away. You know what's happening. And now look again. This is a girl. Do you see her legs? A straw skirt. She's got her arms crossed like this. She doesn't have much of a shirt. I apologize, but I can't find another picture. And we're all grown up, so we can handle it. There's a necklace here. Her head, her hair. There's a face here with the eyes, the nose. It's a completely different, right? See, there's a face here that's not related to the girl. Another face here in a profile. There's a head here on its side with the hair flying out. And another one, you see this profile, another head. Okay, so what's going on here? There are a bunch of different patterns. It's not even the case that the patterns are visible at different sizes. They're all visible at the same magnitude. They're all superimposed on each other. Your brain, first of all, can handle this. So if your brain can handle it, we need to be able to find a way just by copying what the brain is doing somehow, you know. And the thing is, the data, if you think of this as the data set, my job to extract information from the data is to extract the body of this girl and ignore the lion because it's the body of the girl that is relevant to a specific question that I'm asking. So the issue is, so if you look for patterns mathematically, you're not going to be able to tell the difference between this pattern and the pattern of the lion. So you have to talk to the biology and keep paying attention to the question that's being asked so that you find the relevant patterns. By the way, Gene expression microarray data is at least as complex as this. And extracting the patterns is at least as difficult as this. Okay, so this is not an unreasonable, an unreasonable thing to ask. Okay, so let's go to actual data. So I'm going to talk about these two issues, the issue of relevance, which I was just sort of alluding to before, and the issue of the shape of the data. So, so let's look at data from disease. Okay, this is a paper from several years ago. And the idea is, how do you extract from information from tumors the part that's relevant to disease? So let me just say, tumors or tissue has tremendous amount of stuff going on, most of which is not related to the disease. If you take a tumor cell, the tumor cell is doing a slew of things. Most of them are exactly the same as what the tumor cell was doing before it was a tumor cell. And there's variation between one person to the next, to the next, to the next, to the next. So you can't say, let me take everything that's the same in all the patients. And it's nothing like, there's a lot going on. And some of it is disease. So I want to emphasize somehow the aberrant patterns, to decide that certain patterns are aberrant behavior, and then emphasize those so that I identify things that have to do with disease in this data. So the idea is we start with data from disease, and we decompose it mathematically into healthy-like and aberrant. When I say decompose it mathematically, I mean, I don't know why I use that term. What I want to emphasize is something completely different. So I don't mean that certain variables or certain genes are involved in healthy behavior and other separate genes are involved in disease behavior. What I mean is each gene has some behavior that's doing the best it can at continuing to do the job it was doing before when it was healthy, but a little bit doesn't quite make it, or a little bit is a little too much. Okay, so it's sort of like, you know, trying to understand what's happening with the volume of my voice. And suddenly you make the room very noisy. You make the room very noisy, I have to talk louder, not because of the meaning of what I'm saying, but just so you can hear me. But maybe I have a cough, actually I do, so I will talk louder, but not quite loud enough. And so the aberrant behavior, this part of my talking, is going to be a negative thing. Even though I'll be talking louder, but I won't be talking loud enough. Okay, so but the healthy part is how loud I should be talking to compensate for the fact that it's suddenly noisy in the room. So this relies on the following, the method relies on the following very important observation, which is that cells, even when they're sick, still maintain a lot of their behavior from when they were healthy. It's sort of like when you see someone, you see a friend, and suddenly they have the flu, and they walk in, and they have runny nose, and they're wobbling around, and their complexion is somewhere between green and gray, right? They're very, very sick. But you still recognize who it is, right? I mean, and cells are very much like that. So, if you'll pardon this, the situation is not like this. This was a very old horror movie where the mad scientist makes a mistake, and as a result of the mistake, he turns into a fly, basically. That's the plot of the story. And the fly is not, the scientist is no longer recognizable in the fly. That's not what disease looks like. Disease actually looks more like this. If you're not up on pop culture, this is Spider-Man or Andrew Garfield, depending on your point of view. Anyway, and what is he like? He's still Peter Parker. He's still the same guy, but he's got some quirky stuff that's no longer like he was before, so that he can, shoot these strings and he can swing from building to building. Several generations earlier, there was a guy that was swinging from tree to tree. This guy swinging from building to building. That's what disease is like. I mean, the consequences of this change are profound, right? But you still see Peter Parker very clearly in this. So here's quickly the idea of the method. You start off with tissue samples that are from healthy tissue and from the disease, from the tumor tissue. And you generate the data, the microarray data. So these are the round things, means that now I have the data points. And I use the data points from healthy to build a very simple model, mathematical model, for the healthy world. And I'll tell you why the mathematical model is embarrassingly simple. It's actually, there's actually a method to that madness, but you'll see it in a minute. Now you come with data from tumors, and instead of using the original data, you do a geometric transformation on the data, which is a very simple one. Namely, you take the data point, you see how far the data point is from the model for healthy, and instead of working with the data point, you work with the deviation from the model for healthy. So what happens then is you're emphasizing the deviation from healthy. Okay? Now, the model for healthy is very simple. Essentially, it's the subspace in Rn generated by the healthy data, the normal data. Now, immediately, especially in a room full of mathematicians, people will say, maybe do you want to take maybe a manifold a little bit? I mean, do you really think that the world of healthy is a, this entire subspace? And no, it's not the entire subspace, but I actually want the entire subspace. And I'll show you in a minute why. But right now, let's just look at this. So it is a subspace, and I take the tumor data. I drop it. So these are mathematicians, not statisticians in the room. So the language is I take the orthogonal projection of the tumor data onto the subspace, and then I take the perpendicular vector. That's the difference. And so this projection is the normal component of the data, and the perpendicular part is the disease component. In the world of statisticians, this guy is called the fit to linear models, and this guy is called the vector of residuals. It's the same thing. I always use both terms because you never know who's in the room. So let me tell you real quick. Essentially, this is the only work, is how to build a healthy state model. So the normal data points are N1 through Nk. Essentially, I take the subspace generated by these, and then I lower the dimension a little bit. Lowering the dimension, the standard way to do it is principal component analysis, where you basically you turn to zero the directions in which there's very little information. This doesn't work very well for large data, even though it's used sometimes. I mean, and we use it here, but we use it after we do another step. So the reason it doesn't work very, very well in large data is precisely because it's large data. So the idea is that you're going to turn to zero the directions in which you have noise. The problem is that you have so many variables that the data looks as if it's very noisy. It's not really that noisy necessarily, but you have so many variables. Each variable adds more input into these directions that are the noise directions, so that when you try to say, okay, there's signal up to this level, and then beyond this level, I'm going to call it noise because the amount of information drops very quickly. But that doesn't happen for large data. So you can't really detect the directions in which you have signal and the directions in which you have noise with PCA. But what we do is we precede that with a construction that we introduced in this paper, which is basically a smoothing of data construction. Okay? So basically what this does is it brings the points closer together, and the idea is this: instead of working with N1 through Nk, I work with N1 hat through Nk hat, and Ni hat is the orthogonal projection of Ni onto the subspace generated by all the other vectors. So essentially, this is a very simple construction, and what it does is this. It takes the data from Ni, and anything that's unique to Ni, I throw away. Okay? So if it's and the idea is this: I'm building a model here, so I'm not really interested in things that are only visible in one of the data points. Okay, so that's the flap construction. This is just, I don't have time to really go into this, but this is some data simulation to show you that when you use the flap construction, you can identify correctly the correct dimension. So data was simulated. I knew that the dimension had to be 6, but I added lots of noise. And if you use the flap construction, this is essentially a signal-to-noise ratio, and it jumps up exactly at the right point. So then using PCA, I know that I have to reduce the dimension to the first 6 directions. This is what flat looks like, actually. So I have to tell you what these things are. This is a matrix. It's called a heat map. It's a beautiful, very simple, very elegant method of looking at a matrix. If I have a matrix, lots and lots of numbers, I don't know what it means to look at it. I mean, you know, it's just, but there's a very simple trick, namely change the numbers into colors. So you change positive numbers into, traditionally, you change positive numbers into yellow, and negative numbers into blue, and 0 gets changed to black. And then if I also take the matrix and shuffle around the rows so that similar rows are next to one another, and shuffle the columns so that similar columns are next to one another. In other words, I cluster the rows and I cluster the columns. And I look at the matrix, now I see, you know, there's this area that is all positive. There's this area that's all negative. It's beautiful. You can just look at it and you see a pattern in the matrix itself simply because your brain can interpret colors very quickly. So this is the original data matrix. And then this is the flat of that data matrix. So you see how it cleans up all the things. Okay. So, all right, so this is what DSGA does. This is the model. So let me just show you now why, real quick, why I chose this embarrassingly simple model for healthy. Why do I take the whole subspace? The idea is, right, I have the healthy tissue samples. I have vectors corresponding to those. And then I go from there. But actually, that's a lie. This is what it really looks like. So healthy tissue samples are mixtures of many, many different cell types. They're highly heterogeneous. And the data is basically a weighted average of all the different cell types that went in there. And different samples are very, very different in the concentration, the relative concentrations of these things. So now by a very embarrassingly simple linear algebra observation, you notice that somewhere in this large subspace generated by these guys, I have somewhere a vector that corresponds to pure cell type 1, which is some appropriate linear combination of these guys, of the actual data points, and somewhere else, pure cell type 2. Now, I have no idea where those guys are, but I know they exist as long as I don't have very little data. If I have enough data, that's going to be the case. I don't know where pure cell type 1 is and where pure cell type 2 is. But guess who knows where those are? If I have a tumor grows out of pure cell type something. This is a simplified story, but essentially correct. So if I ask the tumor, what's the closest point in this subspace to where you are? The tumor is going to find its cell type. Spider-Man's going to find Peter Parker prior to being bitten by the spider, right? And so the tumor knows where its pure cell type is. So what? So who cares? So here's the point. This is diseased tissue. It used to look like this. What am I measuring here? The extent to which this got messed up. Okay, this is blue here and it's green over there, but yeah. The red part, the disease component, measures the extent to which this particular tissue type was messed up by the disease. So this really emphasizes what went wrong, the pathways that got broken during the disease, as opposed to comparing tumor to some average normal tissue. What that's going to emphasize is that the tumor is cell type 1 primarily, whereas average normal tissue is some weighted average of various different cell types. So that's going to dampen actually the broken part of the disease. Okay. Let me just skip a little bit because I want to show you several things. So the important advantage to this is that the biology that you highlight is different from the biology that you highlight if you don't do the transformation. So in other words, biologists actually learn something new. It's not that you get better p-values. I mean, you do, but primarily the point is they actually learn new biology, which is better biology also. So it's cleaner also at the same time. So this is just a quick example. I want to tell you what these things are. This is called a survival curve. I'm sorry if I tell you things that everybody knows. I don't know exactly what people know, so but I don't spend a lot of time with that. So this is a survival curve. So if you have a bunch of people that are sick, and if some of them die from the disease, you want to actually figure out how quickly they die. And the idea is you draw a plot like this. So I have 4 different curves here. Okay, so let's just look at one of them. You have a plot. The horizontal is time. The vertical is percentage of the total population. And you draw a curve, and every time somebody dies, the curve drops. Okay, so you can see that when people die very quickly, the curve drops quickly. When people die slowly, the curve drops slowly. And you can detect differences between groups of patients like this. Okay? Of course, there are all sorts of complicated issues. For example, if someone has a disease and they walk across the street and a car hits them and they die, you don't want to count them as having died from disease. So they're much more, these little ticks have to do with stuff like that happening. But that's the idea. And these are 2 examples where the biologists did all sorts of experiments to study a specific protein. This one is lysyl oxidase. This one is lysyl oxidase like 2. And they believed that these proteins had something to do with cancer, and they looked at mice and cell lines and all this stuff. And they concluded that, yeah, this probably has to do with the progression of the cancer, but they wanted to know if that's the case also for real people, not just the mice and real tumors. So my job was to look in large cohorts of public data and to see if there is a difference between the patients that have high levels of the protein and low levels of the protein. If you look at the actual data, you don't see a difference. But if you look at VSGA transformed data, you do see a difference exactly as they had predicted. So the difference, so these guys do quite well, these guys do quite badly. But only when you look at the disease component of the data, the transformed data. Okay. So let me now talk a little bit about, would I have about 10 more minutes or so? 7 minutes? 8 minutes? Yeah, so this is the second half of the talk. I'm starting now. A little bit about the shape of the data. So this is a mapper. It's a method that's inspired very much from topology, and it was developed in Gunnar Carlson's group. Gurjit Singh had this as part of his PhD thesis. And it's a very, very beautiful way to associate to a point cloud a shape, a simple graph that you can look at that captures sort of the intrinsic shape of the point cloud. So obviously, if the points are in the plane, I don't need anything. These points are in very high dimensional space. And somehow you you create this graph to see the shape of the data. This is inspired by the construction of a height function on a manifold. And I won't go into that because I'm running out of time, and I'd like to tell you about some concrete applications further down. So the idea is that instead of looking at connected components of the data because you're in a manifold, now when you look at data, you look at clusters, so areas of high density of the points. They substitute for connected components. Now, I'm just going to show you very concretely what this method does. So even if you didn't know about read graphs, you'll still see what's going on here. So that's a data set. You have a function which doesn't have to be a height function. It's just inspired by the way height functions work. And the function, let's say, goes to the real line. And you cover the real line by overlapping intervals, and you take essentially a traveling window like this. And then for each window, you look at the inverse image, you have a bunch of points, you cluster this sliver of the data. So you get 2 clusters basically here, and each cluster is going to be turned into a node for the graph, and you can think of it as a bin that contains points. You move the window a little bit, you cluster now the next sliver of the data, and you get 2 different clusters. And you notice that this bin and this bin, or rather this bin and this bin, have points in common. So if they have points in common, you connect them with an edge. If they don't have points in common, you don't connect them with an edge. And you keep going. And now you see, this is what you see, the points you don't see, the point cloud you don't see, but you see this, and you see 2 gradual progressions. And the colors correspond to where in the line it was. So if there was information, biological information in that, you can also read that off of here. And if the cloud has this kind of shape, you also capture that. And so that's the idea. Notice this is very dependent on the function. If you have a different function, you get a completely different graph. So let's look at how we combine this with data decomposition. So we have this data decomposition of tumor data. We have to find a filter function. These are called, the height function is now called the filter function for mapper. And there's a very natural function to pick. Namely, you have points, and they deviate a little bit or a lot from a healthy state model. So you can just take some measure of the magnitude of deviation from healthy, so basically what we took was just the magnitude. We took several different filter functions, defined by various choices of the norm and powers of that. And this is what you get when you look at breast cancer. Okay, so here are the normal tissues and normal-like tumors. And then there's a gradual progression. These bins are very red, but you can't see them because they're tiny. And these bins are very red, and you can't see them because they're small. Now, I happen to know quite a lot about breast cancer because I worked with breast cancer a lot. So the first thing I noticed was that there is a separation along what's known as the ER status. So ER, estrogen receptor, is a certain protein. And almost any analysis that you do, any reasonable analysis you do on breast cancer, you better see the difference, the distinction between ER negative breast cancers and very ER positive breast cancer. So I breathed a sigh of relief, first of all, because I found that separation. So, because if you don't find it, there's a good chance your analysis is complete gibberish. Okay? And then I looked over here. These are indeed they're called basal-like or triple negative breast cancers. They're a known group. And then I looked at this. Now, there's a known problem. I can't explain why it turns out to be a difficult problem, but it's known that ER-positive breast cancer, some of them do really well, and some of them do really badly. And it's difficult to understand which is which. I mean, what changes between the good and the bad ER-positive tumors. And I thought, oh, my God, these are very, very far from normal, so I bet I found the bad ER-positive breast cancers. So if I look at the survival curve of these guys, I bet it drops down really quickly because they're very, very abnormal. So this is what the survival curve of this group looks like. Nobody dies. That's the curve, yeah? It doesn't drop. Nobody dies. Okay? This is the lesson that, you can have all sorts of theories in your head, and nature says, too bad, I'm doing my own stuff. So what's going on here? Okay, so first of all, there's a lot that's abnormal in these tumors, and yet these people do well. I can't tell you for sure what the biology is, because the biologists have to do a lot more experiments, but I can tell you one possible explanation. Everyone in this room has cancer cells in their body right now. You don't have cancer, but you have cancer cells, and your body finds them and gobbles them up and kills them and gets rid of them. When someone has cancer, the body tries to attack the tumors. That is an abnormal behavior because normally you don't have tumors in your body, right? So it's quite possible that all this abnormal behavior has to do with the body attacking the disease. This is a process that's not well understood, and people are trying to tap into this process because they want to maybe help people with cancer to accentuate these processes so that they attack the tumor. So it's quite possible that there are other possible explanations to this, but this is one possible explanation. Anyway, this is to show you that these tumors, if you use clustering, these tumors get scattered all over the place, but they're 10% of the tumors, so they are an actual group, and this group was not known before. These tumors were essentially unclassified before. That doesn't mean that it's a group. I can randomly choose 10% of of samples and say, oh, look, nobody noticed this group, but they're not a group necessarily, right? So, but it turns out that mathematically the data points are very, very close. They're very similar to one another. And biologically, they make a lot of sense. So it is indeed a group, and it wasn't noticed before. Okay. Do I have 1 more minute? That's all. Okay. So, real quick, this is doing sort of the same thing, but with a much fancier version of Mapper on a different disease, AML, acute myeloid leukemia. And so now there's a company. So this is super fancy Mapper. So this is what the disease component looks like. These are the normal samples. You see that there are 2 groups here. Okay? The coloring is not by position. The coloring is actually by survival. So blue means dying, and red means alive. AML is a very nasty disease, so even the people that have a good version tend to die eventually. I mean, but we all die eventually, but you know what I mean. They tend to die from disease. And you see that there are 2 groups. These guys have the bad form, and these guys have a much better form. And if you look at the survival curves, there's a strong 10 to the minus 6 separation between the good and the bad groups. And now just one more thing. Let's turn things around. And instead of looking at the aberrant behavior, let's look at the healthy-like behavior. Healthy-like behavior, I said before, is not relevant to disease. But actually, it could be relevant to disease. For example, it's not healthy or disease to have light complexion versus dark complexion. If you have light complexion, you're more likely to have skin cancer from a lot of sun, right? But you can't say that your complexion is part of the disease. But it's associated, it can be associated with disease. So we looked at healthy-like behavior for AML. And I'm not going to tell you the details of what we did. But it turns out that AML disease is a disease of the blood, and blood has a very elaborate structure. The cells differentiate and they change. So in the normal space, there's a very elaborate stratification of the data. And essentially, we looked at the tumors, and we defined, in a fairly complicated way, a function from the data to this elaborate structure, and we asked, when the memory of the tumors lands here versus here versus here, does that have anything to do with the disease? And these are the survival curves. It turns out that it has a lot to do with the disease. This is new stuff. I mean, it's new to biologists. So the ones that land early on die very, very quickly. The colors are the same. Okay? And there's a difference between these guys and those guys, and that corresponds to various biological things that we're looking into that make sense, but that were not known before. So that's it. Lots of people participated. I don't want to take your time to tell you their names. Certainly you recognize this person. I think the other people are recognizable more to computational biologists, and of course nothing happens without money. So thank you. I went 4 minutes over time, sorry.